Hello dear students, welcome back to another learning lessons in Physio-Pulse channels. Today we learned about glycogenolysis, the breakdown of glycogen to form the glucose in a simple, easy to understand way. You will find the clear explanations, diagrams, important concepts here. Before we begin, if you find this video helpful, please share, like and subscribe to support the channel. Now let's understand about the glycogenolysis. Glycogenolysis is the biochemical process by which glycogen stored in the liver as well as the stored in the muscles is broken down into the form of glucose molecules when the body needs energy. So this process exactly it takes place into the liver and skeletal muscles, and it is an irreversible process, an independent set of enzymes present in the cytosol which carry out the glycogenolysis. The enzyme that can be degraded by breaking of alpha 1,4 and alpha 1,4,1,6 glycoside linkase. This is the structure of the glycogenolysis, how glycogen is break down to form the glucose molecule. Now to see the process of glycogenolysis step by step. First of all, the action of glycogen phosphorylase enzyme. The alpha 1,4 glycosidic bonds are clipped sequentially by the enzyme glycogen phosphorylase to yield the glucose 1-phosphate molecules. This process called the phosphorylases, and it continues until the 4 glucose residues remain on either side of the branching points, and the glycogen so formed is known as limit dextrin, which cannot be further degraded by the phosphorylase enzyme. Glycogen phosphorylase processes a molecule of pyridoxal phosphates covalently bound with the enzyme. Number 2, action of debranching enzyme. The branches of glycogen are cleaved by the two enzymes activities present on a single polypeptide, that is called the debranching enzyme, and hence it is called the bifunctional enzymes. And alpha 1,4,1,6 glycoside breaks the alpha 1,6 bonds at the branch with a single glucose residues and release a free glucose molecule. The remaining molecule of glycogen is again available for the action of phosphorylase and debranching enzyme to repeat the reaction stated 1 and 2. And the ultimate final step in the formation of glucose 6-phosphate and glucose, that is through this combined action of glycogen phosphorylase and debranching enzyme, glucose 1-phosphate and free glucose, the ratio is 8:1, which are produced. Glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme of phosphoglucomutase enzyme. And the fate of the glucose 6-phosphate depends on the tissue. The liver, kidney, and the intestine contain this enzyme glucose 6-phosphatase that cleaves the glucose 6-phosphate to form the glucose molecule. This enzyme particularly absent in the muscle as well as the brain. So free glucose cannot be produced in these particular tissues. So therefore, liver is the major glycogen storage organ to provide the glucose into the circulation to be utilized by various tissues. In peripheral tissues, glucose 6-phosphate produced by the glycogenolysis will be used for the glycolysis. And it may be noted that the glucose 6-phosphatase is absent in the muscles, and some amount of free glucose molecule is produced in glycogenolysis due to the action of the debranching enzymes. So if you see this exact diagram, then we'll clear that glycogen is that here. It can be with the help of glycogen phosphorylase enzyme, it will form limit dextrin, then to glucose 1 molecule is free, and it will produce glucose 1-phosphate molecule. Ultimately, with the help of phosphoglucomutase enzyme, this glucose 1-phosphate molecule, it will convert to form the glucose 6-phosphate, which will again convert to form the glucose, as well as it goes to the glycolysis also. So this is the debranching or the glycogenolysis breakdown to form the glucose molecules. And if you see the significance of glycogenolysis, the number 1 is it maintains the blood glucose levels. The liver breaks down glycogen during the fasting between the meals, and it prevents hypoglycemia and ensures the continuous glucose supply to the vital organs, especially in the brain. Second important significance, that it provides rapid energy. Muscle glycogen is an immediate source of the energy during the exercise and strenuous activity, faster than the producing glucose from the non-carbohydrate molecules. Number 3 significance is supports the flight and fight responses, because epinephrine can stimulate the rapid glycogen breakdown during the stress or emergencies. So it can fight or flight responses. And number 4, it will help to maintain the metabolic homeostasis like the work, it works together with the glycogenesis to regulate the glucose availability according to the body's need. So the flow chart is that glycogen to glucose 1 phosphate, then glucose 6 phosphate, and then ultimately it will produce the glucose. So students, if you found this video helpful, please like the video, subscribe to the channel, and turn on the notification bell so you do not miss any other next biological lessons. Thank you for watching and see you in our next class.